Okay, so as I was saying, we will start with chapter 1, which is generalities on endocrinology. So in the first slide, you have the different contents in the order that we are going to be talking about them. Chapter 1, introduction, hypothalamic hypophyseal hormone in chapter 2, thyroid hormone in chapter 3, and then the regulation of the calcium metabolism in chapter 4, hormones, insulin while glucagon from the pancreas in chapter 5, adrenal medulla hormones, which are the catecholamines in chapter 6, and steroidogenesis, with the different steroid hormones that are going to be produced from the cholesterol, and their roles and their pathologies. All these are our different chapters. And besides, as I said, if we have time remaining, we will have a chapter about the gastrointestinal hormones, like secretin. So for now, we will start with the principles of endocrinology in our in our introduction. So first, let's start by defining the cellular communication. Of course, when I talk about cellular communication of an individual, I must be talking about a multicellular organism, okay? Of course, it's not a unicellular organism in this case, because there's no communication in a unicellular organism. But here, cellular communication is needed in multicellular organisms because these organisms are composed of complex structure of cells, tissues, and organs. And all these tissues and organs, they do have different functions. But sometimes these organs, they must communicate in between each other in order to accomplish a physiological role. Okay? So take, for example, metabolism of the carbohydrates. I have a lot of organs and tissues in my structure, in my organism, that all are going to play an important role in order to accomplish the function of carbohydrates metabolism in my system. Okay, liver is going to be involved because it's going to store the glucose under the form of glycogen, or give me from the phosphorylase the glycogen into glucose in case of need. The muscle is also going to be playing an important role because it is one of the main tissues that is going to use the glucose when it's going to be active. All our tissues are going to use the glucose anyways. The pancreas is very important as well. And adipose tissue is also playing an important role in the metabolism of carbohydrates. So as you can see, all these tissues are going to be working together in order to accomplish the metabolism of the carbohydrates. So multicellular organisms, they are complex structures of cells, tissues, and organs that has physiological processes such as metabolism, respiration, excretion, movement, and reproduction. Different cell groups of specific functions need to communicate through intercellular communication systems. Okay, so these groups of cells are not independent of each other, but they do communicate through intercellular communication systems in between the cells. Okay, and this communication can be of different types of communication. Now we will see how is this communication can be different in different types. So to have a cellular communication, I need a sender cell where it's going to synthesize the chemical messenger that is going to be sent from the sender cell to a target cell. And it's not like whatever cell that can play the role of a target cell, but a cell to be designated as a target cell must have a receptor, which is a specific interaction between this receptor and the chemical messenger. And most important of all this is not just a receptor to be able to bind to the chemical messenger, because this has no meaning if there is no a functional change that is going to be taking place in the target cell. So the most important thing is at the end a response to the chemical signal. and some changes that should be taking place inside of the target cell in order to say that, yes, this cell communicated with this one. Okay, so I need a response. So for cellular communication, I need a sender cell, and its role is going to be to synthesize and to secrete the chemical messenger. The chemical messenger that by many types of communication might, as I was saying, might in fact be able to recognize the target cell by the receptor, chemical messenger receptor interaction, and most important of all this is to have a response of the chemical signal in the target cell. Now, as I was saying, there are a lot of different types of cellular communications. We can distinguish 3 types of cellular communications with extracellular chemical messenger. Of course, here we are talking when the chemical messenger is not like the second messenger inside of the cell, but it is an extracellular chemical messenger that is going to be secreted outside of the cell. So first is the nervous communication. So this means a synaptic transmission. The second one is the humoral communication, endocrine or neuroendocrine transmission. And the third one is a local communication, paracrine or autocrine transmission. So I will have a slide about each one of these. The first one is the nervous communication between at the synaptic level. So the nervous communication is when you have a chemical messenger molecule that is going to be secreted from a neuron, okay, to another neuron, for example, or at a neuromuscular junctions. Okay, so this is a nervous communication. So this is, of course, a cellular communication because what's taking place in our brain between the different neurons at the level of the synapses, for example, it is a cellular communication. It is one cell that is giving information for another cell, and usually they can be inhibitory or excitatory stimulation. But this is a cellular communication through synapses. And in this case, the chemical messenger that is going to be excreted is going to be called a neurotransmitter. So neurotransmitter is the chemical messenger that is going to be produced by a neuron and received by a neuron, so in the synapses, or by a neuromuscular junctions, as I was saying. So excitation of the sender or presynaptic cell to release a neurotransmitter into a synapse. The neurotransmitter then is going to bind to a receptor, to its specific receptor on the target cell, either a neuron or a muscle, and to activate a cascade of signals to the interior of the cell in order, for example, to have a contraction of the muscle. Okay, one example of the neurotransmitter is acetylcholine, one of the most known neurotransmitters. So لكن هيكون neurotransmitter is a chemical messenger diverse outside of the cell, needs as well a receptor. This is a specific receptor for the acetylcholine that needs to create changes in the postsynaptic neuron in order to continue the message. The second type of cellular communication is humoral system, in the humoral system. So when we talk about the humoral system, is that it's not a synaptic communication here in this case, but then the chemical messenger molecule needs to be taken through the blood. So here where we talk about the endocrine transmission. The sender cell produce and secrete the hormones into the bloodstream. Okay, so in this case, the chemical messengers are going to be called hormones. Sender cell produce and secrete the hormones into the bloodstream. They exert their action via receptor in the target cells. They act at distant target tissues. Okay, so I always need in a sender cell, which is an endocrine cell in this case. And in this case, if it is an endocrine cell, it will synthesize and secrete the chemical messenger, which is going to be called the hormone, into the blood. And then through the blood, it can it can bind to its specific receptor on different types of target tissues that are distant. And this is what is going to be making the humoral system also different from other communications, because it can, in fact, allow this hormone to arrive to many different tissues in our system. Okay, so they act at distant target tissues via receptors, and they are called hormones. Now, I do have another specific situation where I also have a humoral system. يعني my chemical messenger is going to be taken in the blood, but then my sender cell is not an endocrine cell. It is rather a neuroendocrine cell. What is that? That does this mean is that neuroendocrine transmission, I have a neuron that is going to synthesize a chemical messenger and to secrete it. But then this time, it's not going to be secreted into a synapse. Rather, it would go in the blood and to join the bloodstream. So the sender cell are neurons that produce a neurohormone. As an example, we take a vasopressin or ADH for antidiuretic hormone that is going to be produced from in the bodies of specific neurons present in the hypothalamus, and is going to be secreted directly in the blood, secreted in the bloodstream and acting at distant target cells, for example, acting at the level of the kidney. Okay, so here is, in both cases, endocrine transmission or neuroendocrine transmission. I'm talking about the humoral system. This is the system that we are more interested in for our course on endocrinology, in the new endocrine transmission and the neuroendocrine transmission, humoral system. The third type of cellular communication could be from one cell to an adjacent cell that is surrounding it. And here we talk about the paracrine and autocrine. This is what is called the local system. So cells, they also have effects on their neighboring cells. On the cells that are neighboring them, the sender cell produces a chemical messenger, which is going to be called a local mediator. Mainly, these local mediators are growth factors or cytokines or even prostaglandins, if you remember the prostaglandins, which are a local hormones that are going to be derived from the arachidonic acid, prostaglandins and leukotrienes and prostacyclins, playing an important role, if you remember, on inflammation and pain, temperature, all this role of the prostaglandins. They exert their action on proximal target cells, if it's paracrine, or they sometimes can exert their action on themselves if it's an autocrine cellular communication. So local chemical messenger that is going to be produced from the cell and secreted from the cell, but then having its action on surrounding cells or neighboring cells. So these are the three types of cellular communication. In our case, we will be focusing on the type number 2, which is humoral communication, because we are interested in the endocrine system. So this means that we are interested more in the glands in our body, in the endocrine glands of our body, and how they are going to synthesize the different types of hormones and to secrete them into the blood. Now, let's talk a little bit of history about the hormones. Hormones have been discovered long time ago. Okay, this is true and not true at the same time. Why? Because the first ever experiments that have been done and that showed the presence of hormones of an endocrine system have been done in the 19th century. So it's quite a time along for now. But it took them very long time after this, like between 50 years, it was complete silence in this science of hormones before other scientists took over what others have done and complete the story of the hormones. So in the 19th century يعني ما بنحكي بال 1800 نحن 1849 و 1855 here. This was the first ever experiments and done by Berthold on roosters, and that I will be talking about in taking a couple of slides to talk about these experiments, where he discovered the testosterone, but he didn't, in fact, discover the testosterone by itself. He didn't know what is testosterone, but he at least discovered how a chemical messenger or something that would, in fact, change something in the blood and have a very surprising effects on the organism. So we'll talk about this Berthold experiment on rooster by castration. In 1855, Claude Bernard he talked about the concept of homeostasis and the internal secretion and the concept of homeostasis. So he is the one who defined, he gave the definition of the concept of homeostasis and how the organism and our organism is going to maintain our homeostasis independent of the changes that are taking place in the outside environment, what is surrounding us in cold, in heat, in excess, I don't know, eating. If we don't eat enough, we need to maintain a homeostasis in our body, independent of the changes that are going to be taking place. Of course, in interaction with those changes, but in this concept of maintaining constant some variables in our body despite the changes in our surroundings. So this was by Claude Bernard, the concept of homeostasis in 1855. And as you can see, 50 years after, at the beginning of the 20th century, that I have much more now discoveries that are going to be taking place, and a lot of scientists started to be interested in discovering each time and discovering and giving the sequence of different hormones playing a specific role. Oliver and Schaeffer, the first hormone isolated and purified epinephrine from the adrenal medulla. They purified the first hormone. Then Bayliss and Starling gave the definition of a hormone just after them. The concept of neurosecretion, it was done in the 1920s. Neurosecretion, neurotransmitters, chemical messenger of neuronal origin in 1920s, and then followed by the development of sensitive assays such as experimentation, such as techniques, such as the RIA for radioimmunoassay and ELISA, that are techniques used in order to quantify the hormones, for example, or any other proteins if you want. And of course, from the 1980s to 2000, or even to now, our days, the molecular understanding of the hormones has been very developed because of sciences such as molecular biology and the genetics that allowed us to make a lot of changes in our genome and to understand the role of each of our gene and to manipulate, if you want, so we can have animals with a knockout or knock in or knock down. Okay, so we can manipulate, we can decrease, for example, the production of specific hormone or increase its specific production, or knock down a specific hormone in an animal model in order to see its role. And you will see that this idea of knockdown was the very first idea, in fact, that has been done in 1849 by Berthold experiment on roosters that I will be talking about, because he castrated the roosters, he eliminated the testes, and he was looking on the consequences of such an ablation in order to give a role of this organ. So how all these studies on hormones started, in fact, before even Berthold and its experimentation, not only experimentation gave us insights about the role of hormones, but even in the 19th century, يعني كمان بعدنا بال 1800, the microscopists, يعني through the microscopes, were able to describe in detail the tissues and organs, but they didn't have any functional significance for these tissue and organs. So they didn't know the role of each of the organs, despite the fact that they were able to describe them under the microscope. But not only by observation under the microscope, but other observations were done by medical writings, يعني by medicine, early medicine at that time. They used to observe the patient, okay, and what are the changes that they can observe on these patients, okay, and then they would describe them. And sometimes they were able to do clinical correlation between a tissue or an organ abnormality and an observation at the level of the patient, okay, and physiological states. يعني هلا بعطيكم an example of this. So clinical correlations between tissue or organ abnormalities, يعني يمكن يكون في عندي a tissue or organ that has an atrophy or an enlargement, hypertrophy, okay, atrophy or hypertrophy, atrophy أو enlargement, and I can do a connection with physiological state where they were observed. مثلا ناخذ example, if عندي مريض أنا بعدني بالمديسن البدائية وعندي مريض عم يشكي من 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 problem بالregulation of the temperature، عم يبرد كتير أو بيشوب كتير، وبلاقي إنه مثلا ممكن إنه يكون هيدا المريض عنده dizziness أو slowness أو اخذ وزن كمان زياده هيدا المريض. اذا بيجي بتطلع انا as a physiologist على هيدا المريض وعم بتطلع على different organs اللي عنده اياهم، ممكن انه لاحظ انه في عنده hypertrophy بالثايرويد for example، بالثايرويد gland, okay, at the level of the neck here. So in fact I can say that these symptoms physiological state, for example dizziness اللي قلنا عنها أو sensitivity to heat or obesity اللي صار عنده إياها, they are going to be correlated with an enlargement of the thyroid. اوكي بس ما قدر هون الدكتور اذا بدكم او الفيزيولوجيست انه يعرف انه في عندي تي 3 تي 4، انه في عندي هايبر ثايرويدزم او هايبوثايرويدزم، عندي برايمري هايبوثايرويدزم او سكندري هايبوثايرويدزم. بس على القليله عرف نحن كيف عرفناهم هول من خلال الرايتنجز of these medical writings where they were able to correlate لكن some physiological states with changes in the tissues. First experiments, effects of tissue removal resulting in physiological alterations. لكن من وراء هيدول observations they were curious to see what would happen if we do an ablation or we remove excuse me, if we remove an organ or we do an ablation, what would be the effect of this removal on the physiological alteration. And this was what happened first. In fact, when, as I was telling you, Berthold did his first experimentation on roosters. So look at this nice experiment here. We have 4 groups of roosters, 1, 2, 3, and 4. The first group of roosters is going to be taken as a group of control. There's no surgery that is going to be taking place in these roosters, and you can see as a physiological level, the result is that these roosters, they develop comb and wattles in a normal way. They show interest in hands. They have a normal crow. They are aggressive fight behavior, so they have maleness behavior if you want. When Berthold decided to castrate these roosters by ablation of the testes, both testes removed, you can see at the على الفينوتايب في في عندي changes بالphysiological level على الphenotype وعلى الbehavior، مش بس على الphenotype. So the comb and wattles are أول شيء بدي أربط إنه أنا عم بربط شغلي an organ موجود كتير بعيد عن محل الcomb والwattles, which this would be very surprising at that time. Okay, and I see changes in the phenotype of these roosters. Okay, and no interest in the hens. كمان هون عم see I see changes in the behavior. Okay, and the sexual behavior, and they don't fight other males. Okay, so they are less aggressive in their fighting behavior. So أنا عم لاقي changes على level of the behavior وعلى level of the phenotype. So that is very surprising. But he didn't just stop here, Bertolt. He was clever enough to try to reintroduce these organs اللي هن testis by replacing them in the abdominal cavity. Very interesting. يعني رجع ردهم لهيدول التستس باي يرجع يعمل surgery وحطهم بالabdominal cavity، غيرهم مكانهم. Okay, and what happened? The roosters, they had the same phenotype and behavior like the controls. So this shows that in fact the place of these testes is independent for their activity. Wherever they are present, they will be active, and this means that they do not have a direct excuse me, innervation. Okay, يعني they are not very related direct innervation than nerves. Furthermore, he was able to transplant testes from one rooster to another. Okay, ياخذ التستس تبعيدا برجع بحطها in the other one, and same results as before was seen, with the difference in the groups 3 and 4 is that if I replace one testis, okay, for the animal, I will have what I call a hypertrophy. يعني في عندي testes that are larger than in controls, and this is to compensate for the presence of the other testis, is that I have this hypertrophy. So the castration of the cockerels, no development of combs and wattles, fail to exhibit male behavior. These two are the consequences as seen by Bertold. In the replacement therapy in the third group, replacement of one or both testes back into abdominal cavity in either same or different birds، يعني بالثالثة والرابعة الجروبs, normal development of the womb and comb and wattles, male behavior, one transplanted testis larger than normal intact testis, and this is what I call a compensatory hypertrophy. مثل ما قلت compensatory hypertrophy is an increase in size of an organ to compensate functionally for the activity of the other lost organ, because I have one lost organ, so I need to compensate for its loss. What are the significances at the level of this experiment? First, transplanted testes were functional and independent of the nervous innervation. Okay, so if I put them in the abdominal cavity or anywhere else, they are able to be functional. So this means that they are independent of the nervous innervation. The donor organ can be transplanted to another host and remain functional. Berthold's conclusion on these experiments that are very developed for that time, testes secreted something that conditioned the blood. So pay attention to this conclusion, please, where he is saying that the testes secreted something that conditioned the blood, because he didn't realize that the testes secreted a hormone. So there was no hormone word at that time, and he didn't know that he gave an if you want some possibilities, hypothesis, so that conditioned the blood. So it could be that I am going to inhibit something that is present in the blood, to change something in the blood in order to have these consequences. Blood then acted on body of cockerel and caused male characteristics, showed that presence of testes maintain maleness. So at least he knows that the testes are important for the maleness character. Okay, let's say. Now, by the experiments where we have organ transplants or extracts even, this is more even important, extracts. So instead of put the testis transplanted by itself as an organ, after this, scientists started to talk the extracts of the organ, and then later on they started to study these extracts by waste in order to purify the proteins or the chemicals that are present in them and to be sequence them to say what they are composed of. Okay, so the extract as a replacement therapy for absent tissue or organ testis function, activation, transformation of blood constituents. So these are the hypotheses. What this would be. يا اما عم بيصير في عندي activation او transformation of some blood constituents، يعني يمكن يكون في عندي precursor تنقول بقلب الدم وبده شيء من التستس لحتى يعمل له activation او transformation لحتى يصير active. Okay, or a removal of inhibitory substance from the blood. Not only necessary this، بلكي في شيء inhibitory بقلب البلاد وانا عم بعمل له removal by these extracts تبع التستس. or finally which is the real situation, which is the release of hormone into the circulation, and now we know that it is the testosterone that is going to be released and that playing all these roles on the organism. Successful replacement therapy led to purification of physiologically active extracts. Testis extracts could functionally replace testis of castrated animals. Identification of hormonal substances concerned in 1935, very long time after the Berthold experimentation, like about 100 years, a little bit less than 100 years. Testosterone was purified in the crystalline form. So I talked about the first experiment, which is experiment of Berthold, but then many other scientists followed Berthold and were able to do development in the field of endocrinology. Von Mering and Minkowski, they surgically removed the pancreas from dogs. So بعد ما عمل هيك Berthold, a lot of scientists started to do same thing, but by, of course, using different organs. So one, excuse me, they removed the pancreas from dogs, and they were able to see symptoms that relate to the diabetes mellitus in human, such as elevated blood glucose levels, diabetes defects of carbohydrate metabolism due to pancreatic malfunction. So this was a first conclusion انه هل observations اللي عم شوفهم عند العالم اللي عندهم diabetes mellitus it might be related to dysfunction in the pancreas. Then it's in 1912, in the 20th century, that Schiffer was able to name the pancreatic hormone insulin, and then they were able to also discover the insulin in the islets of Langerhans, and that it's not coming from the pancreatic acinia. So here to differentiate between the endocrine and exocrine pancreas, and that to control the carbohydrate metabolism through internal secretion rather than modification of the blood preparation of pancreatic islets injected into diabetic dogs lowered the blood glucose levels. So they discovered the insulin. Sanger, very important discoveries for Sangers in the field of endocrinology, because he got the double Nobel Prize, one for being able to characterize the amino acid sequence of insulin. يعني لحتى يعمل لي sequencing إذا بدكم للإنسولين شو هي الsequence of amino acids تبع الإنسولين. Sanger, and also for developing a very important technique for the sequencing of amino acids and of nucleic acids. So في عنا Sanger method of sequencing that is very famous. and double Nobel Prize for Sanger. And Duvigneau also got a Nobel Prize because he used the Sanger methods to synthesize peptide hormones oxytocin and vasopressin. Later on, we discovered not only the first chemical messenger, but also the second secondary messenger, such as the cyclic AMP. This is represented here. 3 prime 5 prime monophosphate يعني cyclic AMP and الانzyme adenylate cyclase that is able to synthesize the cyclic AMP in the cell, and this was done by Sutherland and collaborators who were able to take broken parts of the membrane of some cells and to activate them through some stimuli to activate liver phosphorylase, enzyme responsible for breakdown of liver glycogen hormones. So الهرمونز حطهم مع الليفر cell membrane عطوني الفاكتور which is the cyclic AMP. So at the time أول شيء سموه الفاكتور, and then لقوا إنه عم يعمل لي activation للفوسفوريليز enzyme in supernatant fraction of tissue homogeneate. So they also define the secondary messenger cyclic AMP involved in the signaling pathways in the cell. After this, as I said, some techniques have been developed, such as the RIA for radioimmunoassay or ELISA, which is more enzymatic. So let me introduce to you these two methods rapidly, because I'm going to run out of battery soon. So radioimmunoassay, it's a technique that allows us to quantify the proteins that are present in our solution, in particular if it is a hormone. Okay, so what you do here is that you have a first antibody that you can see here in black, and you have your antigen that is radioactive. يعني انت نحن مهتمين الانتيجين هون it represents الهرمون اللي انا مهتمه فيها. اوكي، سو لنقول انه هي الانسولين، لنعتبر انه هي الانسولين لحتى اخلي الاكزامبل قريب منكم اكثر. So أنا I have a first antibody against هيدا الانسولين أو هيدا الانتيجين. Then بحط هيدا الانتيجين I need it to be radioactive. So it's like not easy, very easy technique to do. لأنه I need the radioactive antigen. So أنا باخد كمية in excess of my antigen radioactive بسميه hot, and it's bound to an antibody. And then I add my unlabeled antigen اللي هو in blue. This is an unlabeled antigen, يعني هيدا الكولد اللي هو labeled بسميه الهوت. This is the cold. And of course by competition بصير في عندي displacement للred one اللي هو الهوت by the cold one. So by competition I have some of the cold that is going, some of the hot that is going to be displaced for the binding of the cold one. And then I will have to separate the bound from the unbound, يعني بدي أعمل washes إذا بدكم. أو I will precipitate my first antibody by using a second antibody. Precipitate those complexes, antigen antibody complexes, by an anti-immunoglobulin يلي هو بالأخضر هون, second antibody. And I have the bound, this bound fraction. This is explained by steps in this slide. A mixture of radioactive antigen and antibody is prepared. You add to this mixture the unlabeled antigen. The radioactive antigen is displaced by the unlabeled one. The complexes antibody antigen are going to be precipitated or separated by another antibody, and then at the end I can measure the radioactivity in the bound fraction that I have, and this is what I call a tracer. So this is going to be represented by a standard curve. يعني أنا كل ما يكون في عندي الradioactivity أعلى كل ما كان الكولد اللي هو أنا عم بعمل له measurement أقل. طبعا كل ما كان الكولد أكتر اللي هو أنا عم بعمل له measurement كل ما كان التريسر تبع الراديوactivity أقل. As you can see, هول النقط هن my standard curve and this is my unknown here. So in my unknown اللي أنا زدته, I determine the percentage of radioactivity, which is the percentage of tracer in the unknown, and from my standard curve I can determine the concentration of the standard. Of course, again, what I need is it is a nice it's a nice method, but it needs radioactivity, which is heavy to deal with, and it needs of course a standard curve. يعني بدي يكون في عندي أنا انسولين عاملته على 110 1 0.1 and many dilutions for my insulin, for example, to get this standard curve. Another method, which is a quantitative method as well, other than the RIA, it's called ELISA. ELISA is for enzyme-linked immune sorbent assay. Okay, ELISA for enzyme-linked immune sorbent assay. And here in the method is better way. Why? لأنه ما بدها radioactivity. So here I use an colorimetric. This is a colorimetric assay. So this is the difference between ELISA and the RIA. It's a colorimetric assay. Donc أنا هون بعتمد على enzyme that when it's going to be present with its substrate, it's gonna give me some color. يعني البرودكت تبع هيدي الانزيم مع substrate is colorful, بيعطيني color that I can read by spectrophotometry, which makes it more easy for me. So the principle is not very far from the RIA. As you can see here, you need to capture the antigen. This is your antigen that you are interested in. To capture it using a plate-bound antibody. So في عندكم هون plate. Well, the antibody is bound على the plate, and then you add your antigen. Of course, after the washes, you are going to add a second specific antibody, enzyme conjugated. هيدا الantibody is enzyme conjugated. كمان بزيدوا. This is again another antibody. Like this is a method sandwich. يعني من تحت بيرجع الانتيجين بيرجع كمان another antibody with an enzymatic activity. بزيد السبستريت على الانتيبودي, and when I have the color that is coming up, I can measure it by a specific wavelengths على الspectrophotometer colorimetric assay. So what are the differences between RIA and ELISA? RIA measures scintillations by the tracer, as I was saying, due to decay in the radioactive tracer. قديش ما عم يصير عندي decrease كل ما صار عندي أكثر decrease بالradioactivity, كل ما كان عندي لكينا concentration أعلى من my antigen, while إنه in the ELISA measures a color change due to an enzyme tracer. Of course, يعني the reaction بين الانزيم والsubstrate would give me a product which that has a color. The tracer antigen is labeled with an enzyme that catalyzes a color change reaction instead of scintillation fluid. Substrate for the enzyme is added, بدل ما زيد scintillation fluid لحتى أعمل measurement للradioactivity like in the RIA. In the ELISA, I will use a substrate for the enzyme that I'm going to add, and then I will get my product. And the spectrophotometer is used to read the absorbance of light at specific wavelengths to quantify how much of substrate has been converted into the new compound, which is the product, of course. More tracer, bigger shift in absorbance. We use standard curves the same way as in RIA. So هون كل ما يكون عندي الويف لينكس يعني عم بعمل absorbance أعلى، كل ما بيكون في عندي color أكتر، كل ما بيكون في عندي antigen أكتر. بينما بالريا كل ما كان عندي radioactivity أكتر، كل ما بيكون أقل الانتيجين. وهونيك بالريا هو decay of the radioactivity, which means that I have a higher concentration of my antigen. Okay, both cases I do need a standard curve. So this is to give you an idea about the techniques that later on were developed in order to measure هلا كيف قديش في عندي hormones in my blood, for example. These two methods might be used in order to determine the quantitative measurement of the hormones in our blood. Okay. Okay, so I will stop here, and next time we start from the concept of homeostasis, and we have a couple of slides that are very important to understand the homeostasis and how the endocrine and the neuroendocrine integration in the homeostasis regulation. That's what we will be talking about. Okay, do you have any questions? Are you fine?
دكتور
Yeah, one moment just لحتى أشوف. Yes.
دكتور بالنسبة للسلايد اللي فيها الهيستوري بس بنفهم عنها فكرة عامة، ما بنفوت كتير التفاصيل ولا نحفظ أي شيء فيها تواريخ وهيك.
لا اوف كورس التواريخ منا مطلوبه ولا الاسماء تبع الساينتست. اللي مطلوب هو البرتولد اكسبيرمنت، لانه اتس نايس لان كنا اوقات نسال عن البرتولد اكسبيرمنت. You know, like البرتولد اكسبيرمنت نسال مثلا what if you do this the ablation of that organ and you have this consequences, what would be your conclusion. So this is a more analytical question if you want. So this means that what we would like to see, if you understand what type of conclusions you would do if you get some such results. Okay. بس لا اكيد تواريخ مش مطلوبين، اسماء مش مطلوبين، ولا الاوردر هو مطلوب. This is just for you. البرتولد اكسبيرمنت مطلوبه مع الكونكلوجنز يلي انعملوا على اساس هيدي الاكسبيرمنت، انه انا إذا برجع التست to the abdominal cavity, what is my conclusion? It's a connection. And what if I do the extracts وبحطهم هون كمان. هذا الشيء ممكن انه يعني انه في عندي molecule بقلب هدول الاكستراكتس that is going to be play an important role in conditioning the blood. كيف بعرف انه it's in the blood؟ لانه الافكت عم بيكون على distant tissues. يعني انا عم اثر على الكومب، على الوتل، عم اثر على اشياء كثير بعيده بالبيهي، وعم اثر على البيهيفير. هون ببلش بقدر اعمل some conclusions عن الرول لهيدا الاورجان. Any other questions?
دكتورة،
Yes.
أنا لما شرحتي أنت الريا أنا ما كتير صراحة فهمتها. يعني أنا لأي راديو أكتيفيتي بعمل؟ للأنتيجين ولا
مين عم يحكي أصلي؟
أنا
مين أنت؟
يا رفعت إيدي.
لحظة شوي. Okay, anyway. اللي بدي اقوله انه بالنسبه للريا هو نحن مثل كانك عم بدل ما انت عاده عم تعمل ميجرمنت، كل ما كان عندك مثلا انتيجين اكثر، صح؟ كل ما بيكون عندك يمكن ميجرمنت اكثر، هيك معودين نحن. هون الريا هي العكس. ليش؟ لانه انا مبلشه من الماكسيموم. انا مبلشه من الماكسيموم اوف راديو اكتيفيتي، حاطه الهوت راديو اكتيفيتي وعاملتها بايندينج على الانتي بودي، واجاي عم بحط انا الانون عندي عم حطه كولد، سو عم يعمل ديسبيسمنت للهوت. اوكي، سو كل ما يعمل ديسبيسمنت اكثر، كل ما انا عندي انتيجين اكثر بالبلاد يلي عم بعمل له ميجرمنت، كل ما كان عندي انتيجين اكثر كل ما حيخف عندي الراديو اكتيفيتي. هيدا هو البرنسبل تبع الريا.
أوكي
بالنسبه
شكرا.
للانتيجين هو الاورمون. اوكي، بس انت بدك هوت انتيجين وكولد انتيجين. الهرمون يلي انت بدك تعمله ميجرمنت بالدم لحدا unknown, this is the cold antigen. الهوت انتيجين you have to use it لكل العالم. this is your access بتبلش منه.
أوكي شكرا.